Okay, so I want to say just a few words about perception, sensation and perception in general, before we actually move into the visual system. And, sensory detection is carried out by specialized, neurons that are called afferent neurons that have evolved all these very, unique specializations that make them very unlike sort of a classical neuron in some, structural aspects and also functional aspects. So they are able to detect, specific forms of energy, whether that's electromagnetic radiation, for example, within a very narrow range that is detectable by what we call photoreceptors in the eye, or whether we're talking about tactile mechanoreceptors, right, that can, respond to mechanical manipulation. They've evolved all these specialized, kind of membrane, channels and, receptors and, all sorts of specializations. They look very different. We're seeing actually, right, that there's sort of this classical understanding what a neuron looks like that, undergraduates really, sort of take to heart and, really, have the very stereotyped form actually. But these afferent receptors are remarkably diverse in both structure and function. And they're involved in the detection of energy and then the transduction of that stimulus transduction into some kind of neuronal response, right? Which is, typically an action potential. Although we're going to see in the photoreceptor, paradoxically, the absorption, of electromagnetic radiation in photoreceptors is going to paradoxically inhibit, the firing of these photoreceptors, which makes it kind of unusual. But there's a process of stimulus transduction where the specialized afferent neuron, has some kind of structural adaptation that allows it to, grab onto or absorb or, be mechanically manipulated by and transduce that particular energy into a neuronal response, the language of the brain, which is electrochemical. And we talked a little bit about how the way these sensory systems are organized is there's typically a number of discrete, what we call labeled lines, right? Which means like for example, in the tongue, the gustatory receptors that we were talking about, the taste receptors, there's a limited number of subtypes, right? There's like the salty and the sweet and the sour, right? And the bitter and the umami, and the starch, right? There's a limited number because that allows for, fewer genes, right? To code for the production of, these specific labeled lines, for sensory detection and transduction. But then the pattern of activity across those labeled lines allows for a huge diversity of potential, perceptual experiences, because the pattern of activity, differential patterns can then be routed, through different networks to allow for a huge range of potential perceptual experiences. So you might have a nectarine and a peach, right? And there's some similarities in terms of the pattern of activity across those labeled lines, but you don't have to have a specialized peach receptor, right, or a specialized nectarine receptor. You can see those, variations and route them because of that pattern of activity across the different labeled lines, the more limited labeled lines, to discrete regions for, sort of perceptual appreciation and discrimination. This is true also, for example, remember in the emotionally salient inputs, right, through the anterolateral system of the somatosensory system. Remember, you've got the A delta, nociceptors, which respond to tissue damage in the body, and those are myelinated. They send a really rapid, signal back to, mediate that with reflexive withdrawal from injury. And then there's the C fibers, right, which are nociceptors that are unmyelinated, that have a slower, and the A deltas are fast adaptive, right? So they fire initially, so you injure yourself, you pull back, and you also have there's a perceptual quality to that, right? It's a short, sharp versus the sort of throbbing, more persistent, slowly adapting, slower to arrive initially, unmyelinated C fiber. Those are two distinct labeled lines, and yet your perceptual experience of the pain is going to involve the pattern of activity across those labeled lines over time, right, as the signals modulate. And then we also talk about, we're talking about sensory detection and perception the intensity of a stimulus. And sometimes we're going to see this is recorded by the number of cells that are actually firing. So, for example, if you tap lightly versus a real hit, you're going to get a wider, broader, responsiveness from cells, that will let you know it's a more intense kind of an experience. In the ear, we'll see, a sound that's a bigger, amplitude difference in terms of those sound waves is going to move this membrane more vigorously, and more of these specialized hair cells in the cochlea in the inner ear are going to be moved. So that's a population code that can help you to determine the intensity of the stimulus. That's sometimes what happens with that. Sometimes it's the frequency of firing. Like, there'll be faster firing rates. So we'll talk about this in regard to specific sensory systems. In terms of the duration of stimuli, that can, how long a stimulus is present, that can be interesting, right? Because, for example, the Meissners, you touch something and there's the initial firing. It's fast adaptive, right? It fires initially and then sort of slows, right? Versus, the Raffinis, which are involved in, for example, the stretch, the grip, they're slow adaptive, so they keep firing. So generally it's, the length of firing, how long the cells continue to fire, as long as the stimulus is present. But there's some complexity there, obviously, with regards to fast adaptive versus slow adaptive, sensory responses. And then finally, we have stimulus location, right? So there's a famous, biologist named J.B.S. Haldane who is, said many wonderful things, but one of them was, God is a cartographer. Basically mapping is critical. So, for example, if I touch fingertip like right here, right? Well, that cable is going to run in this nerve, and it's going to run through the dorsal root. It's going to run up the dorsal columns, to the brainstem in a very specific location. And that mapping is going to be maintained, into the thalamus, and then ultimately into the postcentral gyrus, the primary somatosensory cortex in the brain. And again, the mapping, the stimulus location aspects are homuncular in nature, right? Where there's an exaggeration, of inputs from, the hands, for example, and the face, versus other parts of the body, because there's more sensory, afferents that are localized in these regions. But there's definitely like a mapping, so that when, for example, if I injure myself, right, that information is going to travel up into, brainstem areas that will allow me to swivel my head, neck, and eyes to the appropriate location for where the injury occurred, because that is all mapped. That mapping is sort of maintained, all the way